Good afternoon, everyone. I'm Peng Fei from Kunming Institute of Botany in Yunnan, China. It's my great honor here today to give a presentation on the variation of plastomes and their potential role in adaptive evolution of pearls. Yes, I will go including the 4 parts, and the first one is a brief introduction to this order. And you can see on the left, this is the APG4 classification, and on the right, this is the plastid plus phylogenomics of angiosperms from our group in Kunming. And of course, you can see the post phylogenetic position in the monocot. Of course, as this is the monocot conference. And this order includes 16 families and more than 20,000 species, and more than 20 species with a global distribution, and you can see all around the world. But however, the species numbers varies greatly among the families from just the 3 to more than 10,000. And we can see this order is the most economically and ecologically important order in the whole flowering plants. It is supposed that the ancestor of pearls may originate in the wet habits, and today there are still many species in more than 10 families living in this habitat, and they also contain all three kinds of photosynthetic pathways, namely the C3, C4, and the CAM pathways. Based on the previous phylogenetic studies, there are 5 major clades. There are 5 major clades in this order, namely such as the basal, such as the xeris, and the gramineas. However, there are many conflicting relationships within the pearls, mainly involving the 4 nodes, such as which one is the first diverging groups, such as the other one is which is the 6th group of the grass families. Here are some examples of different families being considered as the first diverging families. And also who is the 6th group of the grass families. Yes, this is the different delimitation of the xeris clade. So our study tried to solve the conflicting relationships, and we also wanted to know whether the mitochondrial DNA sequence could be used to resolve the to build the phylogenetic relationships, as nearly all of the most studies use the plastid genome sequence. Finally, we wanted to clarify the evolution of the plastomes in Poales. So let's see the second part is the phylogeny of Poales based on plastomes. These are the materials and the methods. We use almost 100 samples with multiple phylogenetics methods, such as basis, as maximum likelihood, and others. Totally, we assembled 11 plastome data sets and 4 mitochondrial ones. They are different. With different genes and different criteria in excluding the poorly augments. For our 11 datasets, we obtained generally consistent phylogenetic results, and the resolution for our focused relationships are like that, such as the early diverging groups, such as the sister group of the grass families. However, we indeed observed many conflicting signals present in the plastomes. For the mitochondrial phylogenetic tree, the early divergent group is different, just with one family. But the sister group of the grass family is the same. There are also many conflicting conflicts with the nuclear and the plastome and the mitochondrial phylogenetics. And we find there are 3 possible reasons for the conflicting signals in the plastome. The first one is the complex plastome structure, such as the very different GC content. And the second is the very different evolution rates. And the third one is such as other biological processes, such as polyploidy and hybridization. We thus decided to further explore the variation of plastomes in Poales. The race variation could be studied from major points, such as the inverted repeats variation, the G duplication, and the G and the intron loss, and the genomic rearrangements, such as the inversion. For this analysis, we obtained 19 plastomes, but 5 of them are not assembled to the whole completed due to the complex structure and the get organelle software revealed. There's also very large variation in the gene size, from 127 base to 225 kilobase. And this one is the second largest reported to date in the whole flowering plants. There's also large variation in the GC content, as you can see in this map, which is parallel to those found in the nuclear genome. This may reflect their adaptation to a variety of habits. And this is inverted repeat variation, and we nearly find all kinds of variation, including 3 inverted repeats, and make this gene very large. And we also find loss of inverted repeat such as and become the direct repeat, the orientation changed. And also like this small inverted repeats, that's only about 2 kilobase. And also this inversion, these inverted repeats were validated by our PCR amplification sequencing. Also, there are many gene loss events observed in different families for a total of 38 genes. And the most frequent loss one is the VCF15. We also find many family-specific inversions, as you can see. And this is the repeat sequence. We find there are few in the early diverging groups, and moderate in the grass family, but there are many in the middle grade groups. And also the same pattern in the rearrangements and also the substitution rate. We then did the correlation analysis and find that the diversity variation pattern of Poales plastome may be caused by the presence of massive repetitive sequence. Finally, we did the selective pressure analysis, and we found several genes were under positive selection, notably along the family that have the C4 photosynthetic pathway and those living in the wetter habit. And this is a brief summary. And we resolved some conflicting relationships within the poles, and also find substantial conflicts within the plastid genomes of the poles, as well as among the plastid mitochondrial and the nuclear data. We also find the plastomic variation exhibit a trait of the, we call it small, large, and moderate during the evolution of the race order. Mainly, this pattern is mainly reflected in the gene size, GC content, and inverted repeat types, and inversions, and among others. And we find that the structural variation is closely related to the number of repetitive sequences. And finally, we provided new science into the complex ethical evolution of plastomes, and this evolution may be associated with their adaptive evolution diversified in very different habits. Okay, that's all. Thank you for your attention, and sorry for my poor oral English. And I think if you are interested in more details, you can send the email to me. Thank you.
Thank you very much. Time for questions.
Congratulations for your presentation. I'm curious about those drawings of the plastomes.
Yeah.
Can you explain them to me? I don't know the subject of your study.
Did you mean the driving force underlying the plastome?
You had in one slide some drawings like secondary structures or something like that. I don't know what it is, but I am curious about it.
Oh, maybe this one. No.
A bit more.
I see.
Yeah. No, just
yes.
one.
Yes.
No.
Let me see.
Back.
I know the slide. Yes. Do you mean there are many circles?
Yes.
How to say? This is somewhat difficult to explain. Yes. This is a result from a software that is used to assemble the plastid genomes. Why they have many very complex circles, I think it's because the repetitive sequence. So there are many possible connections between them. So there would be like that.
Another question? No? No, it seems like there are no more questions.
Okay, thank you again.
Thank you.